This is the poliovirus, composed of an RNA genome and a protein capsid. It causes the disease poliomyelitis via transmission from fecal matter into the mouth. The virus then travels into the digestive system and multiplies within the alimentary tract before entering the bloodstream. This multiplication occurs too fast in the alimentary tract for the body's immune system to respond effectively and outnumber the virus when it reaches the bloodstream. This allows the poliovirus to reach the body cells in the central nervous system and in one of 3 serotypes of the virus cause paralysis. The unanticipated speed of the infection in both the body and in a population is what caused the outbreak of 1916 in New York, as the disease was multiplied and spread too fast to be controlled. The polio vaccine that ended the New York epidemic is one of 2 called the inactivated polio vaccine, which includes all 3 serotypes of the virus, allowing the body to develop an immunity to all of them. The viruses are killed in vitro, which prevents them from multiplying inside the body as mentioned earlier, but still contain the antigens that cause the responses that will be detailed later on. The dead virus enters the bloodstream and body cells of the host, activating 2 responses. The first response is called the humoral response and occurs in the fluids or humors of the body. It begins with the pathogen itself coming into contact with an immature B cell. An antibody on the B cell is a receptor for the antigen on the poliovirus. The MHC molecule attached to the B cell creates a complex that triggers the B cell to multiply, passing the antigen MHC complex with its clones. The 2 types of cells that are produced are plasma B lymphocytes and memory B lymphocytes. The role of the plasma B cells are to create antibodies that attack and kill the pathogen. The numerous methods that the antibodies undertake to do this will be detailed later on in the humoral response. Memory B cells are the overarching name for all of the clones that the immature B cells have created, and their purpose is to be able to develop the same, if not stronger, response in future occurrences of the contact with a specific pathogen and stay in the lymph node of the body for future attacks. The role of the memory B lymphocytes is also to pass on the antigen complex to the helper T cell via its surface protein receptor. This triggers the rapid production of cytotoxic T cells, memory T cells, and suppressor T cells, all of which will be explained in further detail as the process of the cell-mediated response develops. All of these cells contain the specific antigen complex passed on from the B cell via the helper T cell. The role of the cytotoxic T cells is to attack body cells which have already been infected by the pathogens that their antigen complex match, and release chemicals called cytokines which destroy the infected cells. This primary response develops memory B and T cells which contain the specific antigen complex, and when exposure to the poliovirus occurs after vaccination and enters the bloodstream and body cells like earlier, the memory T cells in the form of helper T cells are able to identify it and activate the responses of the memory B cells. It does so through the transmission of cytokines, which also activate the production of phagocytes, which are involved in another component of the immune system. It also induces the production of cytotoxic T cells and suppressor T cells as a cell-mediated response, which will be detailed further on. The memory B cells in the form of plasma cells begin the humoral response and create antibodies which use 4 different methods to kill the pathogens. The first method is the neutralization method, in which the antibodies bind to the antigens and block their access to a body cell, preventing infection. The second method is agglutination, where the antibodies trap the pathogens in a large network that immobilizes them and makes them susceptible to destruction. The third response is called complement activation, consisting of the antibodies activating multiple complement proteins which kill the antibody. The final process of opsonization is where the antibodies tag the pathogens and allows phagocytes to easily locate and engulf them. The B cells also create more memory B cells, which allow them to respond to future infections. The cell-mediated response consists of the helper T cells activating the cytotoxic T cells to destroy infected body cells as detailed earlier. It also creates memory T cells, which allow them to respond to future infections. As both of these responses successfully kill all of the pathogens, it is the role of the suppressor T cells to inhibit the further action of the cells and deactivate them. After the pathogen is killed, the memory B and T cells remain in the body, awaiting to identify another pathogen that matches their antigen complex and repeat the process over and over again. As displayed in the model, the exposure of a patient to a vaccine allows the body to safely develop a response to a certain pathogen that makes it much less susceptible to infection, and is thus an effective procedure which stops the spread of disease.